Good morning, everyone. Hope you're enjoying the conference so far. My name is Prem Mohanty. I am a life sciences product specialist and a product marketing manager at Benchlink. I'm excited to talk about CRISPR gene editing and some of the critical considerations in the associated data management to really accelerate your R&D. Benchlink's mission is to accelerate life science innovation for the betterment of humanity. Our company was founded in 2012 by a team of scientists and engineers at MIT who were frustrated with the poor quality of software they had available to support their science. So with the support from top-tier investors like Andreessen Horowitz, Thrive, Benchmark, and Sequoia, Benchlink built out a range of solutions for life sciences on a single platform. We are fortunate to work with over 500 innovative life science companies, including many of the world's largest enterprises. More specifically, in gene therapy and gene editing, we are proud to work with several leading companies that have helped develop new advances in the field. We are closely aligned with these leading innovators and work in partnership. This has helped build up our domain expertise in CRISPR. CRISPR is one of the biggest breakthroughs in life sciences research. It continues to find new applications in R&D, starting from mechanistic studies and drug discovery to therapeutic applications. Second, CRISPR as a technology continues to evolve rapidly. Scientists are investigating newer endonucleases, novel delivery methods, and improving precision of gene editing, among others. Finally, because of the tremendous potential of this technology, companies are bracing to adopt CRISPR and scale the technology to meet their R&D needs, especially in discovery and screening. If we look specifically at typical teams working in this area, we see several common challenges. Molecular biologists have these newer tools for CRISPR design, but these are often not integrated with their existing software, such as ELN and inventory solutions. This means there is a lot of manual back and forth between these systems. For cell line development or CRISPR delivery scientists, they're working on several different methods or workflows at once and need software tools that can handle both flexibly to accommodate different methods and structure to enable quick setup of multiple similar experiments. With an analytical scientist, the challenge is with stitching together data from multiple instruments, which is a highly manual and laborious process. We think every company and scientific team deserves a solution that will accelerate and support your innovations. When you think about the ideal traits that any digital solution should have in R&D, there are 4 core attributes to consider. First, it has to model the science you focus on, which means having the ability to build data models that fit your science. Second, it has to support the processes and workflows that your team performs and have the ability to scale, iterate, and evolve over time. Third, it has to empower users with data at every level, giving scientists insights about their experiments and team leaders data on their programs. So you can make both scientific and operational decisions quickly with complete and accurate data. Finally, it ensures that users will immediately recognize the value of the solution, see that it saves them time, and adopt it in a way that will help keep them compliant and consistent with the rest of the R&D organization. Let's look at each of these attributes a little more closely. Every CRISPR therapeutic has a variety of entities associated with it, such as DNA parts, genes of interest, guide RNA, nucleases, plasmids, viral vectors, and cell lines. Each of these entities have their own sequences, properties, and ontology. Every resulting gene-edited cell line has experimental results that need to be tied back, not just to the cell line, but also the starting samples and sequences. So this is clearly a data modeling challenge, but it's also an operational challenge, since these also represent the day-to-day functions for lab scientists developing these CRISPR therapeutics. When we think of preparing digital infrastructure for these kinds of challenges, we have to think about both the data management and structure, as well as the operational considerations. Digital solutions have to be adaptable. The IT world has moved away from monolith software and towards more agile approaches. But adaptable shouldn't always have to mean coding and application development. Many life science companies either don't have the resources to do this or want to use the resources they do have to focus on higher value operations. This is an illustrative example of part of a gene editing workflow, and it reflects the constant iteration and evolution that these specialized teams see on a daily basis. So in an ideal world, the data model, the process, and the workflow should be configurable in a low to no code environment so companies can iterate and evolve quickly without being resource intensive or overly dependent on their digital partners. One way we seek to achieve this is with a configurable data model that is editable right from an admin panel. So users can adjust the relationships and metadata on a particular entity quickly and without code. This also applies to process workflows where you can update iterative processes like the example you see here. Once you have the data model and process in place to capture structured data, the next consideration is decision making. A modern digital solution should enable its users to make data-driven decisions. Different members of an organization have different questions that they need to answer and have varying decision support needs. For a scientist, this means uncovering insights across their experiments so that they can design, execute, interpret, and optimize their studies more efficiently without tedious data aggregation and data cleanup. For example, a scientist might be looking at an appropriate cell line to plan the study. Now, instead of emailing other team members or going to separate inventory and ALN tools to look for this information, the scientist should be able to look up and compare expression profiles and inventory data of multiple cell lines by querying a central data warehouse where the data is already standardized during data capture. At the level of team manager, the questions that need to be addressed are at the process and team productivity level. Now, typically, this information is collated and visualized with Excel or other specialized process management tools with separate data entry and reporting workflows. A modern digital solution should be able to connect the experimental and process level information in a single system, such that the managers can track team productivity and throughput easily and answer questions such as number of outstanding requests, assay turnaround time, and view the latest progress on related experiments. Finally, at the executive level, you want a high-level view of your entire pipeline, the ability to compare candidates to make go-no-go decisions and allocate R&D resources efficiently. For example, the executives of a CRISPR therapeutics organization might want to look at key data across lead candidates to identify the best candidate to support major development milestones. Ease of use is a final important attribute of a modern digital solution. Most software tools have a learning curve, and because of poor user experience, a forgetting curve. Only if the solution is easy to use can it ensure users not only get trained, but ramp usage and realize the full value of the solution. An easy-to-use solution allows users to record experiments quickly, tap into the records to uncover the most meaningful results, and finally, aggregate and analyze results across studies from the same UI. This is essential to drive performance, boost productivity, and improve data integrity. Everything that I talked about just now is powered by the Benchlink Life Sciences RD Cloud, which is designed to give you a complete view of your R&D innovation. Benchlink's applications span the R&D process from experimental design, in silico biology, sample tracking to process management. There are 7 applications that Benchlink offers, but they're all offered from a single interface. This makes it easy for users to leverage what they need without leaving the software. All of these applications are built on a common unified platform. They share a configurable data model that maps to each team's unique scientific and operational requirements. Benchlink is built on a modern cloud infrastructure, fully managed by Benchlink, and offers state-of-the-art security and control. And we know that most companies have a rich ecosystem of instruments, software applications, and data stores that their R&D depends on. So we provide an open platform with a full set of REST APIs to move and out-of-the-box connectors for lab automation, as well as ways to move data onto and out of the platform. To illustrate how pioneers in gene therapy are transforming their R&D with modern digital solutions, I would like to discuss Unicure. For those unfamiliar with Unicure, they are a leader in developing gene therapies with several programs in clinical and preclinical development. Prior to Benchlink, they were using paper notebooks and other point solutions such as spreadsheets. This caused 2 major issues. 1, there was a lack of standardization in how experimental details were captured. And 2, difficulty in collaborating with teammates across geographic sites in the US and Europe. With Benchlink, they were able to bring their entire research organization across sites onto Benchlink. The digital tools such as templates and results tables provided by Benchlink helped capture experiments in a standardized manner, which was then searchable and shareable across the entire team. You can find more of these kinds of examples on our website. Thank you for joining me today and have a great day.